Speaker is going to be Dan Coombs from UBC, who's going to tell us about modeling super resolution microscopy.
Okay, I want to thank everybody who invited me here, which is at least Sam, possibly Radek. I've been here for 2 months. I've had a, whoops, where's the mic? All right. Yeah, Sam and Radek and other people who organized this workshop and the program. I've been here for 2 months. I've been having a good time. Don't worry about the EU referendum. Nigel Farage was on this morning and said that on the radio and said that if the UK leaves, then before the UK actually completes leaving, I think it was Netherlands, Denmark, and one other country will already have left. So you can just relax. The EU won't exist. There won't be any EU money to get. And he's never steered me wrong before, right? Apologies. This is again, as is the theme with me at the moment, a fairly experimentally based talk. Okay, but there's a couple of points I want to make. Actually, there's 11 points I want to make. So 11 things that you should know. Now, one of them is less important than the rest. Okay, and that's the first one, which is there's going to be a bit of T cells and a bit of B cells in this talk. Okay, you don't really need, if you're just a casual observer of immune cell dynamics, you don't need to worry too much about it. There are a few people here that probably care about B cells and T cells, but otherwise you can just think receptor and you'll be okay. All right. Okay, so immune cells sense and interact with their environment using membrane-bound surface receptors. So, for example, an immune cell has to have receptors which can bind to foreign things in your body, such as viruses, bacteria, proteins, and peptides derived from those. Okay, but that's how it detects it. That's how an immune response gets started. Engineering these effector cell responses, this is kind of the motivation, is a massive thing. We want to design vaccines, which we're still not very good at doing. We want to have immunotherapies. There's a whole burgeoning world of cancer immunotherapy, and this includes monoclonal drugs and different kinds of cells. So this is actually very important. Well, I mean, everyone's research field is important when you ask them, right? But mine is especially important, okay? Okay, and immune cell surface receptor mobility. So over the past 10 years, and I'll show a little bit of the data for this, is been shown to be the mobility and the spatial organization seems to be tightly integrated with the detection and the response and the signaling. Okay, so this isn't just chemical kinetics. Back in the old days, when I first started, there was a lot of chemical kinetic modeling around, and I mean, it's still there to some degree, but the spatial aspects are extremely important, and that's what I focus on in my work, by and large. And what I'm going to try to do today is tell you about some experiments that have been going on at UBC, jointly between my group and the group of Mike Gold, who's an immunologist at UBC, and unfortunately, this is rather a negative talk. It's going to be about why things are difficult and hard to understand. Okay. All right, so in the beginning, there was confocal. Okay, so confocal microscopy gives you, it's still an extremely useful tool, but it gives you a relatively blurry view of, say, different surface receptors moving around on the surfaces of cells. Okay, so you have this here is 10 micrometers. This is one cell interacting actually with some antigens on a glass. And you can see this kind of blurry picture. You get the sense there might be some clustering, there might be some inhomogeneity there, but they just sort of move around according to this. And then about 5 to 10 years later came TURF. Now TURF was higher resolution. So this is actually the same system imaged in TURF. The red bits here are the white bits here. And what you can see is there's some background here from the cell, but you can see the formation of these microclusters, which are sub-micrometer in scale, and they're moving around on the surface of the cell in response to an antigen. Okay, so basically, around the time that I was doing my postdoc, we had confocal microscope images that were giving us sort of a micrometer scale picture. TURF was somewhat of an improvement. That was around the time that Omer Dushek was in my group in the mid-2000s, and that gave us better resolution. And it's sort of reflected in the modeling, I think. So if you look at 2003 model that I wrote, the T-cell receptor dynamics, well, there's a Laplacian here, right? So what are they doing? They're diffusing around. okay? Which is, in my mind, writing a PDE for these things is kind of the same thing as saying, well, we have a reasonably good idea of the concentration and the concentration gradients, but we don't know where every one of them is, okay? And then in 2009, this is a paper that I wrote with Dodo Das and Oma, it's part of Oma's thesis actually, and you can see here, well, a diffusion coefficient here, okay? Again, this diffusing thing. This is actually a first passage time equation, but it may not be obvious. But the point is that, again, we're modeling in this sort of large-scale diffusion as a reasonable model. And this is a paper here from 2014. There's a bit of mechanics in this paper. But again, you've got this diffusion operator. You've got the sense that things are sort of being blurred out and smeared around. And we have sort of a field approximation. Okay. The problem is that things, when you look closely, really don't look like a field at all. Okay, so the microscopy has improved over the last 5 or 6 years. And this is an image from UBC of IgM, which is a kind of B cell receptor on a resting follicular B cell. So this is a B cell that came out of a mouse. We labeled the receptors on an individual basis with a red dye, and we imaged it in a kind of imaging called storm imaging. I'm going to describe just briefly the super resolution revolution. That was good. So there's a real change that instead of having these blurry micrometer scale images, we have these images which have spatial precision on the order of 10 nanometers or so. Okay. So classically, you can't do this, right? You can't make out details which are smaller than the wavelength of the thing that you're imaging with. And this is why we do X-ray microscopy and electron microscopy, because we want to have things with a high frequency so we can make out precise details. But the photons which come from a point source, such as a fluorescent label, are going to be distributed in your imaging system according to the point spread function of the imaging system. Okay, so technically this should be an Airy function. It's probably more, well, we generally model it as a Gaussian, which is a pretty good approximation. And so what you could do is you could image for a while, accumulate photons which are distributed according to this Gaussian, and the most likely position for your object is going to be at the peak of that Gaussian. So the whole story of super resolution microscopy is fitting and finding the peaks of Gaussians in your imaging data. Okay, but you can only do this if you have a low density of the objects, because otherwise they get blurred together. This is the problem with confocal, right? There's not really much difference between a confocal microscope and somebody doing super resolution in terms of the imaging. It's more in terms of the labeling and then the deconvolution and the finding of the individual objects there. I'm just going to briefly describe 3 methods. Stead, Palm, and Storm, and basically these methods led to the Nobel Prize in Chemistry, was it last year or year before last? Anyway, Stefan Hell and various other people received the Nobel Prize extremely quickly because these methods are really having an impact in biology. So Stead is the one that I argue with people the most about, but basically you have your sample, it's labeled, and you basically you have a donut-shaped laser which removes the fluorescence temporarily from the region where the donut is hitting, and then you excite the fluorescence in the middle. So this is done with chemistry. You have excitation in the middle of the donut, and around it you have depletion of the fluorescence. And what this does, it gives you an effectively much smaller point spread function. You raster this across your image, and you go from this blurry confocal image to quite a sharp image here of some, I think these are microtubules. Okay, you can do this with fixed or live cells. In the next talk, I believe, is that right? You're going to hear about photoactivated light microscopy. So here you have lots of labels which are asleep. You wake them up by gently tickling them with a UV laser. They wake up, you acquire them, okay, and then they bleach out eventually and go dark again, and then you wake some more up and you watch those guys. And you can actually watch them move around, or you can have them stationary as in this image. And you keep repeating until you've run out of labels. What I'm going to talk about most today, although there will be some Stead imaging at the end, is Storm. So the idea here is not so different from Palm. You have fixed cells at high densities. You stimulate them weakly, so you only get a few bright ones in each frame. You stack 5,000 frames, and you go from just a little constellation of dots after 2,500 frames. After 500,000 frames, you have this very nice, precise image. So basically, you're separating the labeling temporally the same as you were in Palm. Sorry. Okay, This is the image which was built in this is an image of the system which was built in chemistry at UBC by Reza Tafte and Keng Chao. And basically the magic is the following, is it has feedback correction for drift. We bought a $250,000 storm system. It's not very good because it drifts. These guys made this for a few tens of thousands of dollars in the basement in chemistry, and if they switch off the feedback, you can see the image, the X resolution drifting all over the place. If they switch on the feedback, they're getting reliable Gaussian peaks down to, they always say less than 5 nanometers. Okay, so basically they're able to image individual objects down to 5 nanometers in X and Y, maybe a little bit more than 5, I would say. This is probably a little optimistic, and not quite as good in Z.
What do you mean by drift here again?
Drift here would be essentially the thing that you're imaging just moving, and you can see that it's not moving consistently. It's wandering around. Yeah. Okay. All right. Thing 4 that you should know. So I explained super resolution. Okay. So we got super resolution. Trends in clustering are not predictable if you slightly change your cell. Okay. So mice and us and mammals have, let's just say, 2 kinds of B cells. They have marginal zone B cells and they have follicular B cells. So you take a mouse, you get its spleen, you can get the follicular B cells out and the marginal zone B cells out. And the follicular ones are kind of sleepy. The marginal zones are kind of awake. All right? And they're big. You can see the difference in size here. These are the marginal zones, these are follicular. The follicular, they don't have much actin dynamics going on. The marginal zone B cells are very dynamic. They're constantly looking for foreign substances. There's a reason for this, is the marginal zone cells are the outsides of the lymphoid tissue. So antigens coming in from viruses and things probably meet the marginal zones first. They're kind of an early warning system. They're sort of pre-activated B cells. This movie's not going to work, but it would show a follicular cell just kind of sitting there doing nothing, and this guy is the marginal zone, and he's probing over the course of a few minutes. He's sending out processors because he's looking for antigen. Okay, and for the benefit of people that like Western blots, there must be at least one here. This is phospho-ERK, which is a measure of signaling, and in unstimulated follicular versus marginal zone cells, you can see extra signaling in the phospho-ERK. Okay, so what are we going to do with these storm images? Well, we're going to try to come up with some ways of comparing them. Okay, so this is my image of IgM labeled on a follicular B cell. And the oldest of the spatial clustering techniques is called the Hopkins index. And basically what you do is you compare the nearest neighbor distances of these dots. This image is made up of dots, of localizations, to random data. And if this is the nearest neighbor distances, this is random. This is the distances of your points to randomly selected points. And so you can see if your data is a spatial Poisson process, then this is equal to a half. Okay? So basically, if you have a half, you have random data. If it's much more than a half, then it indicates some sort of clustering. And I think we can all agree there's some clustering here. So for example, we can generate, whoops, that's better. We can generate histograms of Hopkins indexes for resting cells. So these would be the follicular cells and active cells like the marginal zone cells. Okay, and we can see that, well, this is strange, right? I haven't showed you the picture yet. That was a mistake. But the follicular cells have more clustering. Now, where's the picture? Ah, shoot. Yeah, okay, I should have done this. I should have had this earlier. This is the follicular cell, and this is more clustered than this because this has a lot more, but also there's not much difference between the clusters in the marginal zone cell here. These are zoom-ins. So this zoom in here is the follicular cell. You can see there are gaps, and here there is no gaps. And this is the same plot as before, which shows that the Hopkins index is kind of capturing something here.
What is the size of the cluster here?
50, 100 nanometers. Yeah, sorry. The problem with the software we use doesn't put a scale bar on, but yeah, this whole image is about 5 micrometers across. This zoom in, this would be 1.5 micrometers squared. So the clusters here are 50 to 100 nanometers. Yeah.
clusters of what?
Oh, this is IgM on follicular B cells and on marginal zone B cells. Okay, so IgM is a surface bound antibody, essentially. Yeah. Sorry about the slight confusion there. Okay, next thing in the toolbook of spatial clustering are these things called Ripley functions. Ripley function is, whoops, you measure the number of points within some distance r, and you're going to vary r. You divide by the background density and you normalize. Okay, this is the k function. If you normalize, if you put in random data, you get pi r squared. So what you typically do is use the L function, which is the square root of that thing divided by pi. So again, we're comparing nearest neighbor distances to look for clustering. Okay, and if L of r comes out equal to r, you get random data, which is just a straight, so it's a straight line as a function of r. And what we can see for our follicular cells is there's a peak in the Ripley's L function here, which is whoops, this is the h function, my bad. This is showing, this peak here is showing that the follicular cells for IgM have a typical cluster radius order of 50 nanometers, whereas the marginal zone cells, the clusters are bigger and more dispersed. Okay. All right. We also have played around over the last year or so with measuring. We would actually like to know what the clusters are. So we adapted a method for clustering in weighted graphs called the Markov clustering algorithm. I can explain it to anybody later on if you want. And we're able to also segment the clusters like this. And then we can plot things like the number of clusters as a function of the cluster area. So we draw a convex polygon around the outside and measure the area of that. Okay. Other people have been up to this. This is from a group in France. This is, you take all of your dots and you use these to generate a Voronoi tessellation of your image, okay? And then again, you can measure the number of clusters, say, as a function of the number of dots, so you can do a clustering on the Voronoi elements. One of the nice things about this, is that there are no additional parameters in the algorithm that have to be set by the user, whereas in this method, which we were using, we had like 2 parameters to set. Okay. Just throwing this in, my grad student, Josh, has been working on something which he's calling storm graph because he has a natural ability to do marketing, and we're able to outperform all of the other methods when we're looking at elliptically clustered data, and it doesn't have any input parameters. So for example, we can take this image of an IgM on a marginal zone B cell and automatically generate clusters from that.
Sorry, when
Yeah.
you say outperform, like
Oh, it's just better. I'll show you the poster.
But what's your objective, like?
So how well does it distinguish? So you put random. Do I really have to tell you this? You put random elliptic clusters with slightly different numbers of points down, and it's able to better segment them into the original ellipses. Okay.
What is the goal in identifying the cluster?
I want to know how big they are and how many there are, and how many dots, how many antibodies are in each cluster. Okay, that's what I want to know. Yeah. I mean, it's like I discovered a new animal, right? Start off by measuring weight, height, all these kinds of characteristics. Okay. Okay. All right. So this was the thing that you should know, is that the distinct patterns of that IgM has distinct patterns on the follicular and marginal zone B cells. These cells are not very different, and yet we see quite big differences in patterns. Okay. And we can quantify this using the Voronoi tessellation, for example. So we can find that on the follicular cell we have more or less clusters. There's a typical number of localizations within a cluster on the order of maybe 80 to 100, whereas here on the marginal zones we see that we have small ones and big ones. So there are these small, distinct, we call them oligomers, and then there are nanoclusters which are a bit bigger than that. Okay, 5. And this one is where you should get shot. Okay, maybe I should have put this as number 4 because it's actually more exciting. This is IgD. This is IgM. Now, somebody quickly point out the difference between IgD and IgM. Well, it's up here. It's not even in the membrane, right? So IgD and IgM are very, very similar receptors, and the membrane proximal bit is extremely similar. Okay, I'm not showing the full details here, but take it from me. These chains here are very, very similar on the IgM and the IgD. Okay, in fact, these things are so similar that it's not actually, if you look on Wikipedia, for example, it's not completely clear if they even do different jobs. However, they certainly are separate, they're not clustered together. They don't obey the same clustering. You can just look immediately at the IgD and the IgM on follicular cells, and you can see that they're actually somewhat different. And we can quantify that, for example, by looking at the Hopkins index, or all the other things that I've described. Yes?
Are those different cells, or are those
Yeah.
the same field of view in the same cell?
No, these are these are the same field of view in the same cell. Okay, so this is, sorry, thanks Andrew, that was a good point. This is the same cell. These are not different populations of cells. We double label in two colors. We do storm at the same time on both cells, on the one cell, and we get this is very, that isn't particularly cherry picked. This is the picture we're getting, is the IgD and the IgM are actually clustered differently. And I'll have more on the IgD and IgM in a minute. Okay. On the other hand, if we look on the marginal zone B cells, the IgD, oh sorry, I'm going back and forth. The IgD has the opposite, whoops, where's the logic here? Is IgM on IgM on follicular cells? Yeah, the IgD on follicular cells looks like this. On the marginal zone cells, it looks like this. And this has the opposite trend to what I previously showed you with the IgM. Okay, so we really are seeing differential clustering of receptors where there's minimal difference between the intracellular part of the receptor. Okay, this one's a quick one. Clustered receptors have different signaling patterns. Okay, so what we're doing here is we're labeling phosphorylated CD79. This is the business end of the receptor down here. This is CD79A. We have an antibody against phosphorylated CD79A. So this is a measure of the signaling output of the IgM or the IgD, okay? And on the follicular cells and the marginal cells, we get different pictures on the two different cells. Now this is maybe not such a surprising thing because we know that they're differentially organized, but they're different on the two different cells. Okay. Also, we can do three-color STED, and we can image between follicular and marginal. So this is a very busy slide. I'm not going to attempt to tell you everything here. But there's follicular and marginal cells, IgM, IgD, phospho-CD79A. You do the merge, and we can also look for the overlap, which is in white. And basically, the IgM overlap more with phospho-CD79A on both kinds of cells. So on the follicular cells, there's 30% overlap between red and blue. On the marginal zone cells, there's more, but it's still greater in both cases than the IgD overlap. So again, these receptors, which are different, appear to be which are not very different, appear to be differentially signaling in unstimulated B cells. Okay. Dynamics. All right, that was the end of the statics part. So everything I told you so far is statics. The cell is fixed, we image, we look for clusters. So what about dynamics? These receptors move around. What am I going to say about this? Well, in the beginning, large-scale diffusion was not actually that bad of a model, and everyone was happy. So in the days when everyone just said everything diffused, we were happy. So we could do FRAP analysis of T-cell receptor mobility. So this is an experiment where you label some T-cells. This is a T-cell, and here are the receptors labeled on its surface. You bleach out a spot. The amount of stuff in the reference region here goes down, and then it recovers. And you can fit this with a PDE, a diffusive PDE, and you can obtain recovery curves. And you can find, for example, that the recovery curves are almost exactly the same, and they fit the same parameters between different kinds of T cells. On the other hand, you can mess with the cell using drugs, so you can add cytochalasin or ionomycin, or all two of the things. So, and you find that there's really no difference between the cells by themselves, or the cells with cytochalasin, or the cells with ionomycin and cytochalasin. Only the cell which has ionomycin, which is activating the cell alone, is showing a reduced mobility of T-cell receptors. You can also look at how sort of a natural signaling rather than drug-induced signaling would affect things, and we can see that there's generally a bit of a reduction in the diffusion coefficient from our PDE fit between the T cell clone by itself and the T cells when it's conjugated with another cell which is providing kind of natural signaling to it. Okay, and just to follow up on that, diffusion wasn't bad back then, and it's still not terrible right now. So actually, I have to go quick. But, so if we do a single particle tracking experiment, okay, then we can take images like this. So these are receptors, IgM again, labeled at low density on individual B cells and imaged in turf. And what you can see is, well, they move around. Okay, this is how we do the labeling. We get an antibody, we digest it, and then we put some Psi3 as a fluorescent dye, and these bits will stick to the IgMs and label them monovalently on the B cell. And so what can we do with that? We can take all of the tracks, we can extract them. This is the tracking algorithm extracting the tracks down here from up here. And we can, so here are some tracks from a typical cell. These are some tracks we got from a cell one day. And this is the steps. So this is the step distribution. And it is sort of roughly Gaussian, which is what you'd expect for diffusion. So this is 50,000 or so steps from one experiment. Well, one experiment with multiple cells. And so then we can see, well, how well does, for example, the x-coordinate of the step length correlate with a Gaussian function. So here's my Gaussian fit, and here's my histogram of step lengths over 50,000 steps. And you can see that it's actually not too bad. It does approximately look like Brownian motion should look. Okay, so we're finding steps up to 2, these are pixels, up to 2 pixels, so that's about 200 nanometers. There is a bit of evidence here that there's a population of things which appear not to be taking steps. So are they transiently stuck? It's possible. We might be able to achieve a better fit if we looked for 2 diffusion coefficients where the particles could transition between a stuck state and an unstuck state. Okay. But the problem here, again, is that the trends of the receptor motion are difficult to predict. So, for example, here is IgM on a follicular cell, and here is IgM on a marginal zone cell. And you can see that on the follicular cell, the B cell receptors, the IgMs, are not moving around very much, whereas down here they're quite mobile on the more active kind of cell. So that may not be so surprising. But if you look at IgD, you don't really see the same trend. So here is the difference in the extracted diffusion coefficients from the tracks with IgM. The red line is the follicular cell, so it's a little bit less than, so this is about a factor of 2, than the marginal zone cell. So on the active cell, the marginal zone cell, the IgM is moving around sort of with double the diffusion coefficient as it is on the inactive cell. That by itself is not surprising. However, if we repeat the experiment using IgD, this receptor which is very, very similar to IgM, then we see almost no difference. Like, I wouldn't really report that. That probably is statistically significant because we have thousands of tracks, but it's also probably not really worth saying anything except it's almost the same. So this is generally concerning. All right, the 10th thing is pairs of nearby B cell receptors move randomly, but perhaps not independently. So we were sufficiently concerned about not understanding the clustering and the mobility of individual receptors that we tried to do a 2-color experiment to try to tease these things apart. So remember, one of the problems with super resolution is it's difficult to image 2 things close together in the same color. However, if one thing is green and one thing is red, they can be close together and we're able to image successfully. Okay, so we labeled our resting B cells, in this case, the follicular cells, with red on the IgD and green on the IgM. Okay? And then, well, we took lots of movies, and we define co-localization when the distance between the receptor positions is less than r. Okay? And so obviously if your r is big, then you expect longer and more co-localizations to occur. Okay? So what did we see in terms of duration of co-localizations? So defining 5 different radii, expansion radii here. So this is red and green are within 50, and we count and we see how many frames do we have them within 50 nanometers. How many frames do we have them within 100 up to 250? We see that the duration starting is around 30 milliseconds and increases a bit up to say about 60. It doesn't make any difference whether the cells are activated or not. We can activate them with LPS and it doesn't make any difference here. We didn't try this with the marginal zone cells yet, but treating a follicular cell with LPS is kind of similar. So we can compare this now to the theory. So if you have two diffusing objects, how long should they stay together? Right? We can calculate that. Okay, so the way that we calculate this is with an escape time calculation. So you start a particle here at x, y, and you say, how long is it going to take for it to escape from the disk if it was purely diffusing? So equivalently, I have one particle defined at the center of the disk and another particle away from the center of the disk there. How long is it going to take? It's going to take the diffusion coefficient times the Laplacian of the escape time is equal to -1. This is this Poisson equation which defines the escape time. We can solve that and we get r squared over 16d. So it's normally 8d, but because there are two particles involved here, you effectively double the diffusion coefficient. And you calculate. So we can fit d for the particles without thinking about whether they're red or green. We can just see what d is. And we get a value which is pretty close to 0.1 micrometers squared per second. And you calculate that if r is 250 nanometers, the biggest radius that I considered, then they should stay together for 1.6 milliseconds. And if I go back, well, they stay together for 30 to 60 milliseconds. In fact, if they stayed together for 0.1 milliseconds, we would have a lot of difficulty capturing that because we're imaging at 30 millisecond frame rate. We're actually increasing the frame rate at great difficulty right now to try to get a better resolution on this. But nonetheless, it's still a lot longer than you would expect. Okay. So one way that things can stay together longer than you expect according to Brownian motion, even when they individually appear to be obeying the laws of Brownian motion, is they could have some sort of correlation. So individually they can look Brownian, but together they could be moving kind of in sync. So what I'm plotting here is the dot product between the steps, the average dot product between the steps for different expansion radiuses. So if the dot product on average is 1, that means they're moving in lockstep and they never deviate from parallel. If the dot product is 0, then they're probably on average either moving at 90 degrees or on average moving independently. And what we can see is that in the untreated cells, so the inactive cells, you actually have some correlated movement at the smaller distances, and this more or less goes away. I did check, by the way, that particles which are not within, I made the separation really big, and particles which are not close together actually have 0 here. I get a very tight value on 0 for particles which are far apart. Okay, and with the activated cells, well, there's some correlation, but it seems to be less. And this is something that we can investigate, for example, by treating the cells with other drugs or looking at different cell types. Okay. And let me just remind you that the BAF-treated cells, so the inactive cells, which would correspond to the red plot here, they were showing clusters of about 100 nanometers in the static imaging, whereas the more active cells, this is BAF-LPS, so they have less correlation. They also had, they were the ones, there's something up with this image, but they had kind of bigger dots and some small dots in between. Okay. I just want to comment that you shouldn't go crazy interpreting, I shouldn't go crazy interpreting this because the statistics are pretty poor over here because I have relatively few red and green receptors that cooperated and got close together. If I put the error bars on here, you might not believe this so much. Okay. But I can always take more data, right? I can always take more data. Okay. So the collision between IgMs is modulated, say, by activation or LPS stimulation. Whether or not you apply LPS, you get the same co-localization duration. So it doesn't appear to matter too much whether the cells are stimulated or unstimulated as to how long the red and the green receptors stay together. The range is big, and that's in keeping with the theory for escape time processes. The standard deviation is about the same as the mean. But you do get much longer co-localizations than you would predict, which is indicating that the dynamic data is indicating clustering the same way as the static data is indicating clustering. Okay, we found some weak evidence for motion correlation, so it's possible that when we do a single particle tracking experiment, what we're measuring is not the individual receptors moving around, but it's the clusters of the receptors moving around or interactions between clusters of receptors. And we're still working on this. I think this is a pretty important thing to resolve. Okay. So thing 11, and I guess this is almost the last slide. Thing 11 is statics and dynamics are difficult to reconcile. So I just showed you the only partial success I have so far, and we have to agree it's pretty partial success, in reconciling statics and with the dynamics. So the marginal zone cells show bigger, less well-defined clusters of IgM than the resting cells. They're more dynamic, things are moving around more. However, it's difficult to reconcile this when IgM is doing one thing and IgG is doing something completely different between the resting and activated cells. We also applied, as you just saw, the two-color imaging analysis. This is the good part. But the challenge, I think, for everybody is to combine this into a model which will describe the mobility in keeping with the statics, in keeping with the dynamics, but similarly, and at the same time, it's able to describe the static information when you take snapshots of the model. And so that's something that maybe if I ever talk to you again, I can tell you about. Okay. I should thank the people that did this. The majority of the microscope stuff is done by Livin, and it's analyzed by Alejandra, Josh, in collaboration with Mike and Kang, and Reza is the guy who built the amazing microscope. So thanks for your attention.
So if you think of all the estimation problems that will come up out of all of these things that you're seeing now, like can you estimate correlations between random walks and whatever, how much, I guess it's a dumb question, how much of this already exists in the statistics estimation literature, and how much of it is new that should be done because you saw these things?
That's a good question. I mean, there is a good literature on correlation functions. The ecologists, where's Scott? Scott could probably say what the ecologists have been up to. So they label elk and watch them move around. The problem the ecologists have is they don't have very many tracks. So you label an elk and it walks around, okay? But certainly, my benefit is I can take as many thousand tracks as I want. It just takes 1 day per 10,000 tracks. But in terms of the estimation, the field has suffered from tracks being expensive and therefore people over-interpreting what they have. So there are methods out there which give you far too much result for far too little data.
Let me rephrase.
Yeah.
The statistics people that do these estimations, they could have studied such problems, but maybe have not had the reason to study them. That's more what I'm asking you. Let's say, are there people that have done correlated diffusions, not because it had to do with receptors, but because, oh gee, they wanted to write a statistics estimation paper?
Yeah, I'm not aware of that particular problem being answered.
Okay.
Yeah, we always have a look. And the statistics literature can be a bit impenetrable to people like me, but yeah.
So question, you had the correlated movement between these receptors, and you had the sometimes correlated and sometimes anti-correlated clustering, and I lost track with which. So is there any consistency between the ones that are moving together and the ones that are clustering or not clustering together?
Okay, so tentatively, the ones which cluster in the 100 nanometer clusters appear to be moving together more than the ones which are not so, the clusters are not so well defined or are generally bigger. Yeah, that's what I would say.
So that's consistent.
It is, that bit is consistent. Yeah, that's why I described that as my partial success. Yeah.
And the follow-up, is there any chance of recapitulating this in vitro, or is it just, is all the motion intrinsically connected to the cellular apparatus?
Yeah, so if you treat the cells with cytochalasin, for example, you will typically get different results with these kinds of experiments. In terms of hope, yeah, there would be hope if Petra Schwiller decided she wanted to do this, and then it would happen, right? There's no hope at UBC because we don't have those kinds of in vitro systems going. Yeah.
So what is information in the size and the stability of the cluster in terms of the signaling. Is it important to have a small cluster that it is stable for a long time, or we need a big cluster because this is an important piece of information for the cell?
Yeah, the dominant model is that the clusters are small and stay separated until you activate, and then the clusters will merge together. With that said, it's hard to reconcile all of this with the differential mobility of similar receptors, or differential clustering of similar receptors on the cells. We also have some information on other signaling molecules, so we can label anything we want and correlate anything we want here. So for example, if we label CD45, it would have been, according to that model, CD45 is an important signaling molecule, it's on the surface. According to the model I just said, which is that the clusters basically glob together when they start signaling, the CD45 should have sort of come in with the clusters, but it really doesn't in our hands. So, big question as to whether that is the correct model or not. But that's the biological model that's out there.
So you showed that very similar receptors kind of have different patterns. Is there any thoughts on how is that?
I have no idea. As far as I know, the cytoplasmic tails of these, not cytoplasmic tails, the bits of them in the membrane and the cytoplasmic tails are almost the same. So really, it's a very subtle thing as to how the control is being exerted, and how does it know to keep the IgMs and the IgDs kind of separated? This is a serious question I don't know the answer to.
Can you separate the correlations from hydrodynamics from those from binding?
Sorry, can you say that again, Steve?
You separate correlations from hydrodynamics from those from binding? Because you'll have hydrodynamic interactions, I'm guessing, too, being as well, +5.
Oh, yeah, so you mean distinguish interactions between two receptors which are close together, direct interactions, versus there's a geography of things which are moving them around together.
That's right. There's a plan if one diffuses this way because of lipid interactions, they'll go the same way because of flow interactions.
I mean, statistically, okay, there's something I can think of. Well, there's only one thing I can think of is you could look at the fluctuations of the distances between them, and that might give you a clue. Although even my concern with that would be the following: is you would as soon as you reduce your confinement radius that you're looking for, you get fewer and fewer of these things. So even when we have 10,000 tracks, I only find 20 which are within 50 nanometers for more than one frame or something like that. That's why I was saying statistically it's a little bit dubious right now. So that would be tricky. What I think is a better experiment to do is the following: is label in red at high density and try to track clusters using turf. Simultaneously label in green at low density and try to track the correlation of the motion of the individual green guy against the background of the red guy. That would be the experiment that I want to do, and we think it's feasible. So maybe later in the summer that will happen. Yeah.
Can I assume there's only one storm label per receptor?
Yeah, that's a great question. I took that out of the talk. No. And we're actually interested in, we basically have a hidden Markov model for the number of labels per receptor and the number of receptors per cluster. You get to observe only them when they switch on, but they might still be sitting there dark. And we're trying to do a hidden Markov process. It's quite interesting because it has a time component of they switch on, they switch on, they switch off, they switch off, and simultaneously a spatial component of where you localize them. So you've got two pieces of information to put together into your model. And we're working on that right now to try to figure out how many labels per cluster, given that some labels might switch on and off multiple times. But yeah, that's a great thing.
Do you have an idea how many there are?
Well,
2, 10?
No, labels per receptor. No, it won't be more than 4. It'd be more or less impossible for it to be more than 4. Yeah, that's a constraint actually in our optimization. Yeah.
So I'm just kind of curious, presumably they cluster because they have to interact with each other. And I just wondered if you could resolve the distance between the receptors in a cluster. How close is that?
Well, I can tell you the distance between the peaks of the localization Gaussians.
Yes.
Yeah, and they're as close as we can measure.
So 5 nanometers or
So
less.
5 nanometers. Some of those might be multiple fluorophores on the same antibody attached to the receptor, as was just pointed out. Yeah. So certainly, one of the beautiful things about storm data is you can make the dots on the screen as big or as small as you want, right? So you can make it look incredibly crisp, or you can make it look a little blurry. I think the most honest thing is to have them be dots of the same radius as the standard deviation of the Gaussian, which you also get to estimate from the data.
Just a comment. One, if you have diffusion and there's correlations and you want to study them in, let's say, the hydrodynamics, they do have, that does have a signature in the fluctuations, but it's best to look at a non-equilibrium. For example, if you could dye half of the cell only, and even at high density, it doesn't have to be storm, and then look at the fluctuations as the thing spreads by diffusion, you will see a difference when they're correlated versus when they're uncorrelated. I mean, we've done stuff like that with hydrodynamics, and you do see the signature of the hydrodynamic correlation. But if you just did fluctuations where the whole cell was dyed, then you wouldn't see it. It would look Fickian, but when you make them half the cell died, then you see some signature in statics.
So this is a kind of a frat type thought. Yeah. Thanks.
Okay. Thanks, Dan. So we have so many talks. We have